All right, so let's start with homeostasis first. So, let me just mute you all. Yeah. All right. So, homeostasis is the maintenance of constant internal temperature, and we maintain homeostasis to keep the set point stable within the narrow limits in our body. So, homeostasis is important because we want our body to maintain a constant set point. All right. And homeostasis happens because of negative feedback mechanism. In this, you have a stimuli. A stimuli is an internal external change in factor which is away from the normal set point. So, for example, if our body temperature is 37, it suddenly goes to 50 degrees. Then this is a stimuli. This is a stimuli, and a stimuli is detected by something called receptors. A receptor is a cell, tissue, or an organ which detects the stimuli. And there are multiple kinds of receptors. The most famous one is a thermoreceptor. There is also osmoreceptors. Osmoreceptors, from its name, detect water points, and thermoreceptor, it would detect the temperature of our body. So after receiving this receptor, the receptor detects the stimuli, and the coordination center consists of tissues which receive, process, and message the messages in the form of hormones or nerve impulses from the receptor and determines the appropriate response and sends it to the effector. So the coordinating center consists of different nerves, tissues, and also our brain, as well as different hormone systems. And the job of this coordination center is that it will process and manage this stimuli that is taken from the receptor, and it will make an appropriate response. And this response will then be forwarded to the effector. And the effector, from its name, effector will receive this message from the coordinating center, and it will carry out a correct response or a correct reaction. And a response is the reaction carried out by the effector. And because of this response, the body goes back to the normal or set point. So there are two types of feedback that you need to know for homeostasis. The first one is positive feedback. In positive feedback, the response reinforces to the original stimuli. This is very uncommon. In positive feedback is very uncommon, and the response taken by the positive feedback is intense. It intensifies over time. For example, labor pain. When you're giving birth, right, the body will constantly increase the pain that you'll receive. This is an example of positive feedback. So the the stimuli, the effect would increase over time. The pain received due to labor will increase or intensify over time. So this is positive feedback. In negative feedback, the response counteracts the original stimuli. And this is very common in the body. And this is important to maintain homeostasis or stable environmental internal temperature. And the in negative feedback, the response happens so that you go back to the set point. And for example, if you have glucose levels, if the glucose level increases in the body, then the body counteracts this response so that the glucose level decreases and goes back to normal. But if it decreases a lot, then it would increase back by secreting insulin. And this will go on until you have the normal set point. And the normal set point in this case is the normal sugar levels. So yeah, we'll talk more about insulin and glycogen, but yeah. So let's move on to excretion. Excretion is the removal of unwanted products of metabolism. And these products are toxic or poisonous to the body, So it can cause damage to our tissues. And there are four types of toxic products that you need to remove from the body. The first one is obviously carbon dioxide. And carbon dioxide obviously comes from aerobic respiration, and it's excreted via our bloodstream. Whereas urea is a nitrogenous base, and it is made in the liver from excess amino acids. And again, uh, these three products, urea, uric acid, and keratin, all of these three are excreted by the kidneys. Excreted by the kidney. The carbon dioxide is the only product that is secreted uh, via the bloodstream. And so, yeah, and uric acid, remember, is also produced in the liver like urea, and it is produced due to excess purine bases of nucleotides. So, uric acid is caused by excess purine, whereas urea is caused by excess amino acid. Remember the difference. And again, uric acid is also excreted by the kidneys. Keratin is also made in the liver, and it comes from the keratin amino acid. Keratin amino acid, and it's also excreted via the kidneys. So yeah, remember these four waste products. So wait, what is this? Yeah, so let's talk about urea more, because urea is the main excretory product found in our urine. So urea is the main nitrogenous excretory product. It is formed from excess amino acids, and it is made in the liver cell, because liver uh, breaks down uh, our proteins, our unneeded proteins back into amino acids. When we have an excess of this amino acid, then we'll get urea. So the first step in making urea is deamination. Deamination is when you remove an amine group and a hydrogen atom from an amino acid. And this produces something called ammonia or NH3. And remember that NH3 is a base. So it can, if it accumulates or it adds up, then this base can change or have an effect on the pH, because it will react with H plus ions. And this can cause changes in pH. And we don't want change in pH in our body, because change in pH means that our protein structures will change, because we have hydrogen bonds in our protein structures. So excess ammonia can accumulate, and this is toxic. So to prevent this, we go through something called a urea cycle. A urea cycle is when the excess ammonia that accumulates, reacts with carbon dioxide and makes something called urea, which we know. And then this urea is excreted by our kidneys. And so yeah, and this is the structure of our kidney. You have to memorize this structure. Let me zoom in. So the outside of our kidneys, is called the kidney cortex. The kidney cortex is this layer which is outside of the medulla, and the medulla are these egg-shaped organelles. You'll see these egg-shaped or fan-shaped. This is called the kidney medulla. And the renal artery is the blood vessel that takes the blood inside the kidney. And remember that a renal artery will uh, be taking blood away from the heart into the kidney. So the blood is going away from the heart. The blood is being pumped away from the heart into our kidneys, and this happens due to renal artery. Remember that arteries always carry blood away from the heart. And the renal vein is the blood vessel that carries blood from the heart back into from the kidney back into our heart. So remember, renal vein carries blood from the kidney to our heart. And remember that veins always carry blood towards the heart. Uh, the arteries always carry blood away from the heart. And we'll talk more about this in the coming classes. But yeah, for now, just know renal artery and renal vein and their jobs. And so let's move on. Renal pelvis is this, it's present inside of these egg-shaped organelles. Let me just erase all of this. Yeah. So yeah, renal pelvis is the inside of these egg-shaped or fan-shaped. So renal pelvis is present inside of the medulla, and the ureter is the tube that carries the urine produced in the kidney out from the kidney into the bladder. And the job of this bladder is to store urine. And yeah, and the urethra is uh, the job of urethra. It's a tube that carries urine out from the bladder. So also you have to know the pathway of urine. So first, the urine is secreted from the ureter. So the first the first pathway of urine is the ureter. Ureter carries the urine from the kidney to the bladder, and this is the place where the urine is stored. And from this bladder, the urethra is the tube that carries the urine from the bladder to out of the bladder. And yeah, so out of the bladder, urethra is the job. So remember, first is ureter, then bladder, and then urethra. All right. Yeah. So let's move on. The structure of kidney. Uh, Capsule is a tough protective layer that has three main regions: the cortex, the medulla, and the pelvis. So this capsule right here has this renal capsule. The empty space is called the renal capsule, and it has three main components: the cortex, the medulla, and the pelvis. And we'll talk about each later on. So nephrons are tiny tubes in kidneys present in both the cortex and the medulla. The cortex and medulla are the main components that contains these nephrons. And a nephron can be broken down further into five different structures. The nephron, right, has the main component, the Bowman's capsule. The Bowman's capsule is present at the cortex. The proximal convoluted tubule, this you'll see in the exams as PCT. It's the short form. The proximal convoluted tubule is found at the cortex. The loop of Henle is divided into two. So this right here, this is the loop of Henle. It's divided into two. One part is found in the medulla. One part is found in the medulla. The other part is found in the cortex. So the loop of Henle has two parts. One is the descending limb and one is the ascending limb. The descending limb is the limb that goes down, and this descending limb is found in the medulla. Whereas the ascending limb is the limb that goes up, or it ascends. And this part is found at the cortex as well as the medulla. So this is the ascending limb. And after the loop of Henle comes the distal convoluted tubule. The distal convoluted tubule is this small tube right here, and it is found at the cortex. You have to know the differences between the distal convoluted tubule and the proximal convoluted tubule. Remember that the distal means it is further away from the Bowman's capsule. So distal convoluted tubule comes at the last part of the nephron. So the last part of the nephron after the distal convoluted tubule is the collecting duct. And the collecting duct is the place where all of the urine will be stored. And this collecting duct, remember, is found in the medulla. So collecting duct is found at the medulla. So yeah, in summary, you have to know the components as well as the places where it's found. So yeah, remember all of these places. So why am I explaining all of this? Because of something called ultrafiltration. Ultrafiltration is when you filter out small molecules, including urea, out of the blood into the glomerulus. And from the glomerulus, you pass on this blood into the filtrate in the Bowman's capsule. So let me show you the diagram of the Bowman's capsule. This right here, this entire thing is called the Bowman's capsule, and you can see it's found here as well. So the Bowman's capsule has two main arterioles, the efferent arteriole and the efferent arteriole. The efferent arteriole's job is to take blood in, and the efferent arteriole's job is to pump blood out. And remember that when the blood enters this glomerulus, the inside of this Bowman's capsule is called the glomerulus. And remember that once the blood enters the glomerulus, the blood will be filtered out. So all of the unwanted stuff like urea and keratin and all of that stuff will be filtered out. And what would remain, the thing that comes out from this efferent arteriole is blood that is important to us, that we actually need. And the filtrate is the stuff that the glomerular filtrate is the stuff that contains all the unwanted stuff, and this filtrate will flow along the nephron. So how does the glomerulus filter out the blood? Remember that the blood in glomerulus is separated from the lumen of the Bowman's capsule by three main layers. So there are three main features that will filter the blood out. The first one is the endothelial of blood capillaries of the glomerulus. The endothelium of the blood capillaries of the glomerulus has many filtration gaps. These gaps allow filtration to happen. And the second type of filtration mechanism is the basement membrane. The basement membrane is made of two things, the collagen and glycoprotein fibers. And these basement membrane act as the main selective barrier for filtration. So remember, the basement membrane is the main selective or filtration barrier. And this is where the main filtration will happen. And finally, the third type of filtration mechanism is the epithelial cells of the Bowman's capsule. These are also called the podocytes. And the podocytes have finger-like projections that form gaps or filtration slits. So podocytes, remember, they form gaps that act as filtration slits. The gaps help in filtration. All right. So a summary, how is the glomerulus structured for ultrafiltration? Number one, it has large gaps in the capillary endothelium. And also there are filtration slits formed by the podocytes. And this causes easy movement of substances from the blood plasma blood plasma into the lumen of the Bowman's capsule. And also, another way filtration happens is due to the diameters of the efferent and the efferent arterioles. Remember that the diameter of the efferent arteriole is much wider compared to the efferent arteriole. What do I mean by this? Where is the, yeah, right here. Let me just erase this. The efferent arteriole, the efferent arteriole's diameter is much wider compared to the efferent arteriole. So this one, the efferent arteriole's diameter is much smaller. This means that large amounts of blood will enter, but only small amounts of blood will get out of the efferent arteriole. This causes a lot of blood to accumulate in the glomerulus, and this will increase the blood pressure in the glomerulus. This causes filtration to happen. So more filtration will happen due to the glomerulus. Let's move on. Where was I? Yeah. Moving on, this leads to high blood pressure in the glomerulus, then the Bowman's capsule. So fluids are forced out of the glomerulus into the Bowman's capsule. And finally, we have the basement membrane, which acts as the main filtration slit. And the basement membrane will prevent red blood cells, white blood cells, and large plasma proteins from passing through this filtration. And what is the filtrate? So this filtrate right here, the filtrate that we form in the basement membrane after it passes. So this filtrate will contain these things. The filtrate contains, it doesn't contain any large proteins. What it does contain is water, amino acids, glucose, urea, inorganic ions such as sodium ions, potassium or chloride ions, uric acid, keratin, and some vitamins. So yeah, you have to know the composition of this filtrate. And the glomerular filtrate passes through these gaps between the podocytes and into the renal capsule. And from here, it will combine with the urea to make urine. And then this urine will go through all of that, the urethra, and then the bladder. All right. So yeah. So after ultrafiltration, after the blood is filtered in the glomerulus, then comes all of this stuff. Proximal convoluted tubule, loop of Henle, all of this stuff comes after the blood is filtered in the glomerulus. Remember, the glomerulus or the basement Bowman's capsule is where the filtration happens. And yeah, so what comes after the Bowman's capsule is selective reabsorption. So selective reabsorption is reabsorbing all the essentials from the filtrate back into the blood. So remember that the filtrate contains important stuff, nutrients that we need. For example, vitamins, glucose, water, all of this is important to our body. So this will be reabsorbed back into the body through selective reabsorption. This happens in proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting duct. So selective reabsorption starts in the proximal convoluted tubule. This is the main site for glucose, amino acid, vitamins, and chloride reabsorption. And remember, the walls of PCT are made of a single layer of cuboidal epithelial cells. Cuboidal epithelial cells. And so the first step in selective reabsorption is the active transport of sodium ions. This happens in the proximal convoluted tubule cells. I'll just say PCT from now on. So this sodium ions are actively transported from the PCT into the blood, into the bloodstream. So first step, sodium ions from the PCT move into the blood by active transport. This happens through a sodium-potassium pump. And the concentration of sodium ions in the PCT cell would start to decrease because now Na plus ions will move into the blood. So we'll have less Na plus ions present inside our blood, inside our PCT cells, because there is high concentration of Na plus ions in the blood now and low concentration of Na plus ions. So the second step is that Na plus ions in the PCT of the lumen. What is the lumen? The lumen is this structure that is present before the PCT. So the Na plus ions in the PCT lumen will now diffuse down the concentration gradient from into the cell lining. And this happens via facilitated diffusion via a co-transport carrier protein. So I told you guys that Na plus ions moves into the blood. This means that the PCT cells will have a lower concentration of Na plus ions. And so the lumen, right? This right here is the lumen. This lumen has high Na plus concentration. And so the Na plus ions diffuses from high concentration to low concentration from the lumen into the PCT cells via facilitated diffusion. And remember, facilitated diffusion happens through transport proteins or co-transport proteins. Carrier proteins are transport proteins. So this lumen and PCT has co-transport proteins. And when the sodium ions move into the PCT cells, it is transported along with glucose, amino acids, and vitamins. And all of this is present in the lumen. Remember, the the lumen the lumen stores all of the glucose, uh, the vitamin, and the amino acid, all of that stuff, and it is co-transported along with sodium ions into the PCT cell. And after that, once the glucose, amino acids, and all of that stuff is co-transported with the sodium ions, the last step is glucose, amino acids, and all of that stuff will diffuse into the blood via transport proteins, via facilitated diffusion. All right. So let me just erase all of this and explain it in the diagram. So remember, step one is potassium ions, sodium ions from the PCT diffuses into the blood. Sodium ions diffuses into the blood. This causes a decrease in sodium ion concentration in the PCT. Because of this, the sodium ions present in the lumen of the PCT will diffuse into the PCT. So sodium ions diffuse into the PCT cells via co-transport proteins. With the sodium ions, the nutrients like amino acid, glucose, vitamins are also transported. And the third step, the final step, is the nutrients, the important nutrients like glucose, amino acid, vitamins are transported into the bloodstream via facilitated diffusion through transport proteins. These are the three main steps of selective reabsorption that happens in PCT. So, all right, is this clear till now? You have to be very clear with all of the process. All right. I'm assuming it's clear. So let's move on. Remember that glucose is all actively reabsorbed into the blood. There is no glucose present in the urine. Generally, this is the case. But when you have glucose present in the urine, when glucose is present in the urine, this means that you have a problem with your kidneys or you have diabetes. Because when you have diabetes, there is a lot of glucose present in the blood. So the blood does not need any more glucose to be reabsorbed. So because glucose is not needed in the blood, glucose will be found in the urine. So a symptom or a diagnosis for diabetes is that you'll have a lot of glucose present in the blood, in the urine, sorry. So yeah, you can test the urine out through different tests. And if glucose is present in the urine, a large amount of glucose is present in the urine, then you'll have diabetes. All right. And so yeah, let's move on. Amino acids, vitamins, and chlorines are also actively reabsorbed. Um, Water and urea is passively reabsorbed. That means only some amount is reabsorbed. some amount is reabsorbed. Uric acid and creatine are toxic to the body. When you have large amount of uric acid and creatine, these are toxic. Because of this, none of them are absorbed into the blood. So creatine and uric acid are actively secreted from the blood into the PCT, into the lumen. And yeah, so it is found in the urine. All right. And yeah, so how are PCT cells adapted for selective reabsorption? Number one, they have uh, a large amount of microvilli. This means that you have large surface area, and large surface area means more reabsorption. Second, it has a lot of mitochondria present, because remember that there are a lot of transport proteins that need active transport. Active transport of potassium ions into the blood from the PCT. Remember, PCT into the blood, the sodium ions from the PCT into the blood is going active transport. And active transport requires a lot of energy. And mitochondria will make a lot of energy. So you have large amounts of mitochondria present in PCT cells. After that, there is high infolding of basal membranes facing the blood capillaries. And finally, this also increases the surface area. This helps in the surface area. And finally, there is presence of different transport proteins in the membrane. For example, you have co-transport proteins, you have pumps and aquaporins. We'll learn more about aquaporins later on. All right. So let's move on to the loop of Henle. Once the blood passes through the PCT cells, it will move to the loop of Henle. And here you'll also actively reabsorb any essentials. So the loop of Henle is located in the medulla as well as the cortex. It is mainly used for water reabsorption. There are two parts to loop of Henle. The descending limb. The descending limb is permeable to water, sodium, and chloride ions. And the ascending limb is impermeable to water ions, but it is permeable to sodium and chloride ions. So remember, ascending limb does not allow water movement. Descending limb allows water movement. So how does water get reabsorbed? In the ascending limb, remember, in the ascending limb, sodium and chloride ions will move out of the tube. So the sodium ions, chloride ions move out of the tube by active transport and into the tissue fluid of the medulla. Remember, medulla is empty space. This is where all of the sodium and chloride ions will be transported. When the chloride ions move out of the loop of Henle, this causes the water potential, the water ions, uh, to increase. And when you have chloride ions in the medulla, remember, when you have a lot of chloride and sodium ions in the medulla, this causes the water potential in the medulla to decrease. So water potential decreases. So water, the you have high concentration of sodium and chloride ions in the medulla. The renal fluid, this is the fluid that is present in the loop of Henle. This becomes more dilute. And because water ions, remember, water potential, water ions will start to get reabsorbed. Uh, Water ions will diffuse from high concentration to low concentration. So high concentration is present here, low concentration is present in the medulla. So water ions will move out of the cell of the loop of Henle and move out of loop of Henle, and this results in the renal fluid becoming more dilute. The renal fluid in the medulla becomes more dilute, and the remaining fluid in the loop of Henle then enters the distal convoluted tubule. All right, so the steps for reabsorption. Number one, sodium ions and chloride ions move into the medulla. This causes low concentration of water in the medulla. So water moves out of the loop of Henle from high concentration to low concentration, low water potential. This is osmosis. All right. So osmosis happens, water moves out from the loop of Henle, water gets reabsorbed. All right. I want to clear out one thing. Uh, where is the diagram? Right here. No, this is not the diagram. Yeah, this is the diagram. Remember that the collecting duct is the duct where all of the urine will be stored. All right. So the job of all of this, all of the fluid that is present here is not needed by our body. So the PCT, the DCT, the loop of Henle, the collecting duct, all of this contains components of urine, the stuff that we don't need. So all of the stuff that we need is actively removed from this system. So, all right. Um, yeah, this is how water is reabsorbed into the body. And yeah, so uh, moving on to collecting duct. Collecting duct is found at the medulla. The tissue fluid of medulla has high concentration of solutes. Remember that once the fluid enters the collecting duct, the collecting duct has high concentration of solutes. Solutes can be ions and all of that stuff. So water will move out of the collecting duct from high concentration to low concentration. And there is high reabsorption of water back into the blood. And this causes the formation of urine in the collecting duct. You don't have to know a lot about collecting duct other than the fact that urine is produced at the collecting duct and it's stored at the collecting duct. And all of this is happening, remember, it's happening at the kidneys. And water is being reabsorbed into the blood, right? And the rate of water absorption, the amount of water that is absorbed in the collecting duct and the loop of Henle is controlled by an hormone called ADH. ADH stands for antidiuretic hormone, and this is osmoregulation. Osmoregulation. All right. Osmoregulation is the control of water movement in the kidneys. Yeah. And we'll talk more about osmoregulation in the next class because this is a very long topic. All right. So, yeah. If everything is clear till now, I'll see you tomorrow. We'll discuss osmoregulation in the next class. All right. Yeah. So, see you tomorrow. Bye.